Okay, in the last video, we finished talking about the physiology of muscle contraction. So how an actual muscle contracts and all of the different proteins and steps involved in that. And so now we're going to move on in this video and talk about some kind of additional topics related to muscle activity. Okay, and so we're going to start with talking about muscle tone. All right, and so muscle tone is just the idea that at any given time you have, sets of muscles that are active and sets that are inactive. And this is particularly important for muscles that help to maintain posture. And so you can have different muscle groups and motor units within muscles that are activated at a certain time, and then they can switch off, and that helps to prevent fatigue for muscles that we use a lot, like abdominal and some of our back muscles to help maintain posture. All right, so it's kind of the idea that motor units, all right, trade off to maintain posture. Okay, and that helps to prevent them from fatiguing. Okay, good. All right, and that maintains kind of a consistent muscle tone because you have these individual motor units turning off and on and kind of taking each other's place. All right, and so then with muscles, they can experience states of being hypotonic. All right, so this would be a state where a muscle has become very weak, right? And so this would be associated with a decrease in muscle tone. All right, and one time you could see this is with flaccid paralysis in particular. All right, and so if a muscle is flaccid, that just means that the muscle has gone limp, right? It's not functioning, it's not contracting. With flaccid paralysis, that muscle is limp because there's damage to the peripheral nerves. And so we're going to learn more about peripheral nerves with the nervous system, but this is just referring to nerves outside of the brain and spinal cord. So these would be the motor neurons going directly to the muscle itself, right? So this would be damage to the motor neuron. Okay. All right, so kind of on the flip side, you can have a muscle that is hypertonic, all right? So this would be more of a muscle spasm. So instead of being weak and flaccid, not functioning, this muscle would be spasmed and in a state of kind of almost constant contraction. And so we can see this with something called spastic paralysis, all right? So when a muscle is spastic, it becomes rigid. Right, versus a muscle that is flaccid is very limp. Right, and with spastic paralysis, this usually results from damage to the central nervous system. So this would be the brain and spinal cord. All right, so central nervous system. I'm just going to abbreviate that. Wow, there we go. Nervous system damage. Okay, and so the reason this causes the muscles to spasm is because when you damage certain parts of the central nervous system, that's the brain and spinal cord, you lose inhibition on the muscles. So things that tell the muscles, not to contract at certain times. All right, and so then they spasm, right? So loss of inhibition. All right, and so the brain functions, we're going to talk about this in a lot more detail when we talk about the brain, but the brain, one of its big functions with muscle contraction is to kind of mold raw motor signals, right? That says, to like all of the motor units in your thigh muscles contract, it kind of scales them, it pares them down, inhibits some, and that sort of thing, so you can have finer control and more coordinated movements. So The brain is doing that function. So if you damage, certain parts of the brain or the spinal cord that's carrying that information, you can lose that inhibition and get spastic paralysis. Okay. All right. And then additional terms you may have heard of before are muscle atrophy and muscle hypertrophy. All right. And this actually affects the muscle cells themselves. So muscle atrophy, the muscle cells, you don't lose muscle cells. However, each muscle cell loses myofibrils. All right. So loss of myofibrils. All right, and so this loss of myofibrils and atrophy are typically associated with the muscle not being used. All right, so a lack of use. If somebody's bedridden or paralyzed, you can see this. All right, so lack of use. If somebody's in a coma for a long time, their muscles could start to atrophy. And so lack of use / nerve stimulation. Okay. And if this atrophy is prolonged, it can become irreversible. And so the actual muscle cell, if you have prolonged atrophy, can be replaced with fibrous tissue. So you lose that cell entirely, and that's when atrophy becomes irreversible. All right, so then hypertrophy would be the opposite of this. So you're increasing the number of myofibrils, right? So increase the number of myofibrils. And so this would be when you are working out, right? You're using your muscles more. So like if you're lifting weights, that sort of thing. So this is from excessive muscle use. All right, this would be things like weightlifting and that sort of thing. All right, because generally for your muscle to respond by adding more myofibrils, what happens is when you lift weights, that excessive amount of weight, you're actually tearing those little myofibrils a little bit. They get damaged, and then when they repair, your muscle cell responds to kind of compensate and make sure that doesn't happen again by adding more myofibrils. All right, good. So that's hypertrophy. So the muscle cell itself will increase in diameter, right? So this would increase the diameter of the muscle cell. All right. Okay. So now we're going to kind of shift gears, and we're going to talk about ATP as it relates to muscle activity. All right, so obviously muscles need energy in order to contract. So let's start out by pointing out there's 3 points in what we talked about in the last video where that muscle cell specifically needs ATP. Okay, and so the first thing we talked about, all right, we pointed it out, very specifically, is that the muscle cell needs ATP for myosin to release from actin. All right, so that's one. All right, so myosin release from actin. All right. Also, you need the that same ATP, right? So we're not going to make this a separate step, but the same ATP that releases myosin from actin is broken down in order to cause the power stroke, right? So that's kind of one use because it's the same ATP, all right? Okay. And then in order to have an action potential, you need the activity of the sodium potassium pump. Because remember, we talked about with action potentials that change in the charge of the membrane is caused by sodium rushing from the outside of the cell to the inside of the cell. All right, and so the reason sodium can do that is because you have this sodium-potassium pump constantly working to put sodium on the outside of the cell. All right, so this is needed for action potentials. Okay, so that thing is constantly running, and muscle cells have a lot of them because, you know, they need to be able to have that action potential travel all the way down the muscle cell. Okay, so that's our second requirement, step that requires ATP. And then our third step is going to be kind of at the end when you're ready to stop muscle contraction. Remember, we put the calcium back into the sarcoplasmic reticulum. All right, so you're taking calcium from where there's less and you're putting it into the sarcoplasmic reticulum where there's going to be a lot of calcium. So that is active transport. All right, so active transport of calcium into the sarcoplasmic reticulum. We're just going to abbreviate that SR. Okay, so putting that calcium back into the sarcoplasmic reticulum requires ATP. Okay, so where does all this ATP come from that the muscle cell is using? Let's talk about those sources. So we're going to go through them kind of in order of how your muscle cell prioritizes using them. Okay, so your muscle cell's first source of ATP is just ATP floating free in the cytoplasm of the muscle cell. All right, there's not much, there's very, very little. This would last maybe a few seconds of muscle activity. All right, once it's gone through that source of ATP, it then resorts to creatine phosphate, all right, or phosphocreatine. You may see it written both ways. It's just referring to the same thing, okay? And so what this is it's a molecule of creatine, all right? So that's the C part here. And then it has a phosphate bonded to it with a high energy bond like this, all right? And so what can happen, what muscle cells do is they transfer this phosphate with a high energy bond to an ADP. All right, so we're going to add an adenosine diphosphate. All right, and we're going to take this phosphate and we're going to add it on. Okay, and then that is going to make ATP for us. Okay, so then you just have free, right, creatine would be floating around without its phosphate. Okay. So your muscles can do that as a kind of quick source of ATP. It really only lasts about 15 seconds or so, right, of muscle activity. So it's not a long-term source of ATP. All right, so we'll say it lasts about 15 seconds. Okay. All right. So then your muscle cells obviously are going to need a much longer lasting source of ATP. And so that's where we get to using glucose for ATP. Glycogen is stored in the liver. All right. Remember, glycogen is the storage form of glucose. Okay. So storage form of glucose. It's made in the liver and it's stored. And so when your muscle cell needs energy, it signals to break down that glycogen into those individual glucose molecules. Okay? And so then from there, that glucose can be broken down into, and you'll learn in lots of detail about this in A&P2 when you do metabolism. We're just going to speak very generally. All right, to use this glucose, the first thing you have to do to it is essentially break it in half and make 2 pyruvic acids. All right, so this glucose has 6 carbon molecules. Each of these pyruvic acids only have 3. So you're basically breaking the glucose in half. You end up with pyruvic acids. And then what you do with those pyruvic acids depends on if oxygen is present or not. Okay? All right. So the process of making this pyruvic acid, all right, so we take our glucose, we break it down into 2 pyruvic acids. You end up also making 2 ATP in that process. This is called glycolysis. All right, and it will happen whether oxygen is present or not. Okay, what you use those pyruvic acids for is going to depend on the oxygen. All right, so let's talk about if there's no oxygen present. That is anaerobic respiration. All right, so no oxygen present. Okay, and so in that case, that pyruvic acid is going to be broken down into lactic acid, which is just a waste product made during anaerobic respiration. And so you end up with just those 2 ATP, all right, that you made during glycolysis and the 2 lactic acids. All right, so this is a waste product. You would just get rid of that. Okay. So going the anaerobic route, no oxygen, you're only getting 2 ATP for every glucose molecule that you break down. All right, so let's look at the oxygen route. So this is with oxygen. That is aerobic respiration. So with aerobic respiration, you can go through the rest of cellular respiration. That's what the process is called. And this happens in the mitochondria of the cell. All right. In the mitochondria. And it's this long process. Again, you're going to learn about all the steps in A&P2 with metabolism in the digestive system. But what you end up with is you end up making 36 to 38 ATP and some other byproducts like carbon dioxide and water and heat are made as well. But you make a lot more. Okay? So going this route, this aerobic respiration route, is more efficient and it provides a longer term source of ATP. So for every glucose molecule, you're getting 36 to 38 ATP versus anaerobic, you're only getting 2. Okay. All right. One is not necessarily better than the other. It's just that aerobic respiration provides a more long-term source of ATP versus aerobic or anaerobic on this side without oxygen. Provides less, but it's quicker. It's much faster, right? So it's a faster source, but it doesn't last as long. Okay. All right. And so this process, obviously, all right, the deciding factor between which way you go is if oxygen is present. So where can that oxygen come from? All right. So we probably all know that blood transports oxygen, specifically the hemoglobin in our blood cells are going to transport oxygen. All right, they pick it up from the lungs and they take it out to the tissues where it's needed. Skeletal muscle cells also have a specialized protein called myoglobin. So this is a protein that can bind to and hold on to oxygen. All right, so that it binds and it basically like stores oxygen. Whoops. And stores oxygen. And it's found in muscle cells. All right, that's actually why muscle cells look red, because they have all this myoglobin, and when myoglobin is bound to oxygen, it has a red color to it. And so it's got like these, it means that the muscle cells have these stores of oxygen that they can just use very readily to go through that aerobic respiration. All right. Okay, and so your muscles vary based on, they're different based on their preference between aerobic respiration and anaerobic respiration. So some muscles can prefer to go the anaerobic route, and some muscles can prefer to go the aerobic route. And so we'll categorize those as slow oxidative muscle fibers and fast glycolytic muscle fibers. So the slow oxidative muscle fibers, right? If you can just remember oxidative, meaning they like to use oxygen, all right? And aerobic respiration is slower, so they're slow, okay? So these muscles prefer aerobic respiration. That means they're going to need plenty of oxygen, so they have a very good blood supply. They're usually a little more red in color because they have a lot of myoglobin as well as that rich blood supply. And obviously for doing cellular respiration, they need a lot of mitochondria. And one other way to recognize these cells, so they're going to have a rich red color, right? But they're also very thin in shape, right? So they're much thinner than their fast glycolytic counterparts, okay? And so this would be something like a long distance runner, all right, or cross country runner. They would have a lot of slow oxidative muscle fibers, all right, because they need to make ATP over a long period of time, and they use it a little more slowly, right, because they kind of pace themselves to be able to travel that long distance. Okay, so let's compare that to our fast glycolytic. All right, so they prefer glycolysis, right? So this would be that anaerobic, all right, without oxygen route. So just using the ATP made during glycolysis. Obviously, those muscle cells are going to have fewer mitochondria. They don't really need them because they're not going the aerobic route as often. They are going to be very fast at making ATP because it's just the one step to make those 2 ATPs. So they make it very quickly, but they also fatigue very quickly, meaning they run out of ATP. All right, so run out of ATP. Okay, and these muscle fibers physically look thicker, right? And so you would see this in a sprinter. And so if you ever compare, like during the Olympics or something, when they have these different events going on, compare someone who is a sprinter and what physically their muscles, their musculature looks like to a long distance runner. Sprinters have very bulky muscles, all right? That's because they have a lot of these fast glycolytic fibers. And long distance runners have much leaner muscles because the actual muscle cells themselves are much thinner. Okay? All right. So those are differences in the ATP kind of production and usage of different types of muscle cells. And so now let's look at kind of the activity of muscle cells, right? So the actual contraction of muscle cells. So we use a myogram to measure changes in tension in a muscle cell. All right, so what we're looking at here, would be one muscle twitch. All right, so the response of a muscle to a single action potential from its motor neuron. All right, so on this graph, we have time on the x-axis, and we have tension on the muscle on the y-axis. And so this little red arrow here would indicate where that action potential was being sent. Okay, and so with a typical single muscle contraction, a single muscle twitch, there are going to be 3 important phases. All right, and so the first phase is called the latent, or period is called the latent period. All right, it's very small. It would be right about here. Okay, and so it's this time, we have our action potential here. It's this very short period of time between when that action potential is sent and when the muscle actually starts to increase its tension. Okay, so this is the latent period. Okay, and so it's only a few milliseconds. It's very, very short. And so this is the time between action potential and an increase in muscle tension. Okay, so it's just that little kind of phase before that muscle starts to actually contract. All right, so then from there we have the period of contraction. All right, so that would start at the end of the latent period and go all the way to this peak here before it starts to travel back downwards again. All right, this is called the contraction period. And so during the contraction period, muscle tension is increasing. Muscle tension increases. And then the last phase we have, all right, would be, I'm going to kind of draw it down here, all right, from here to right here, and that would be the relaxation period. All right, so relaxation period. And so this is just the time when muscle tension is decreasing. Okay. All right, so muscle tension decreases. Okay, so those are the three kind of periods or phases of that muscle twitch when you're looking at a myogram. And so now we're going to talk about a couple kind of unique concepts that relate to skeletal muscle and one that relates to cardiac muscle and their contraction. Okay, so the first concept is the idea of a refractory period. Okay, and so this is the time, the period of time that right after the muscle is stimulated by an action potential, it cannot be stimulated again. Okay, so after, we'll say, yeah, so time after muscle stimulation, when the muscle cannot be stimulated again. Okay, so in skeletal muscle, this is a very, very short amount of time. It's like 1/1000th of a second. All right, cannot be stimulated again. Okay, so you can stimulate with the skeletal muscle pretty quickly. You can send series of action potentials to skeletal muscle. This is going to be different when in ANP2 you're learning about the heart. They have a much longer refractory period, which is important for cardiac muscle, heart muscle, because it needs to contract and then be able to relax before it is asked to contract again. Because if you think about the heart, when it contracts, it's forcing blood out, all right? And then it needs to completely relax and fill with blood again before the next contraction. So the muscle in the heart is specially designed to make sure you have that longer refractory period so that the heart can fill with blood. That's not the same with skeletal muscle. You can stimulate it pretty quickly back to back. And we'll talk about kind of what happens when you do that. Okay, so the first thing that we can talk about is summation. Okay, and so with summation, this is when you stimulate the muscle before it's completely relaxed. All right, so before it completely relaxes, goes back to baseline. All right, and what happens is that you get a stronger contraction the next time, right? That next stimulation. Next contraction is stronger. You're kind of adding on to that previous one, right? That's why it's called summation, is stronger. Okay, so let's look at this graph, right? So we stimulated, our first stimulation was right here at the very beginning, all right? So we get our contraction period. It starts to enter that relaxation period here. Before it can actually reach all the way to the bottom down here, we stimulate it again. And so now we get another period of contraction that is much bigger than our original contraction. That is summation. So if you stimulate a skeletal muscle an additional time before it has a chance to relax, you'll get a stronger contraction the next time. That's called summation. Okay? And so then also with skeletal muscle, we can get a concept called tetanus. Okay? So with tetanus, rapid stimulation gives you sustained contraction. All right, so somebody actually who is suffering from tetanus, like you can get from a rusty nail or something like that, what happens is your muscles become overly stimulated. You keep activating those receptors and the muscles just basically spasm. All right, so this would be sustained contraction. All right, and so muscles would lock up in that case. But the idea of tetanus is very common, right? So you know if you are holding something, right? So if you are, let's say, carrying your anatomy textbook, right? It's kind of a little, heavy, all right? So those muscles in your biceps that are helping you hold up the weight of that textbook, those are in tetanus, right? They're in sustained contraction because otherwise you would just drop the textbook. So tetanus is a good thing, right? Unless it comes from actual the tetanus toxin, and that's not something you want. But you use tetanus all the time with your muscles. And so what it looks like is you have these rapid action potentials being sent like this. And so the idea is you send another action potential before you even reach a point where you're starting to relax, right? So you just keep that muscle in that constantly contracted state. All right, and so you can sustain that tetanus as long as your muscle has the ATP to remain contracted. Okay. All right, and then our last concept for these myograms is trepe. And now trepe, I want to be very clear, is only seen in cardiac muscle. Right? So it's only in cardiac muscle. So heart muscle. So it's a very special characteristic that heart muscle has. Okay? And so what happens with trepe is that the muscle can contract and relax, and you stimulate it again just as it relaxes, and you get a stronger contraction. All right. So we contract and relax. And the next contraction is stronger. All right. Next stimulation causes a stronger contraction. Okay. All right, and so you can see that here. So this muscle it received its action potential, it contracts, completely relaxes, you stimulate it again, and then your next contraction is stronger, right? Higher tension than the original, right? You stimulate it again, and your next one gives you an even higher contraction, okay? And so this relates to the presence of special calcium channels in cardiac muscle cells, and so they have the ability to add more calcium into the inside of the cell, which helps to make the contraction stronger. All right, so So this is because more calcium is available. Okay. All right. So that's trepe. Again, trepe specific for cardiac muscle, right? Not seen in skeletal muscle. Okay, and so then there are two types of contraction. So one is isotonic contraction. So with isotonic contraction, you would have a constant amount of tension. All right. And then the muscle is shortening. All right, so shortens. Okay, so an example of this would just be like you're lifting weights. So you have tension, right, constantly, kind of consistently increasing on the muscle, and that causes that muscle to get shorter, okay? Versus an isometric contraction, all right, we have an increase in tension, but the muscle does not shorten. All right, so muscle does not shorten. So an example of this would be if you just go to like a wall, right, and you try to push as hard as you can on the wall. All right, the harder you're pushing, the more you're increasing the tension on your muscles, but those muscles never actually get any shorter, all right, because that wall is not going anywhere. Okay, so that would be isometric. All right. Okay, so that's the last topic for this video. However, in your chapter review for this chapter, at the very end, there are two sections, one on cardiac muscle and one on smooth muscle. And those are meant to help you kind of review cardiac muscle and its structure and kind of think critically about what you've learned this chapter about skeletal muscle and how is that skeletal muscle different from cardiac muscle in its structure and kind of in its function. So there's kind of questions that'll guide you through that for cardiac and smooth muscle. So make sure you go through those, fill those in. If you have any questions about them, just send me a message. I'd be happy to help. All right. But I think it helps if you do that kind of on your own independently. You'll learn a little more and try to actually really think about it. Right. So like one question, for example, is do cardiac muscle cells have sarcomeres? All right. So what do we know about cardiac muscle cells? Well, we know that they are branched cells. They have striations, right? They have a single nucleus. They're connected to each other by intercalated discs. Okay, so does any of that apply to what you learned this chapter about sarcomeres? And so then your brain should, eventually make the connection. Well, yeah, right? They're striated. And so those striations relate to those I and A bands we see in a sarcomere. So yeah, likely they do have sarcomeres, which yes, they do. Okay, so do that as you go through those two sections, right? And try to fill in those questions as you're studying. Okay, and so then our next video will go over the actual muscles and the names and kind of what they do and their functions.